And we're rolling.
All right. So the point of this lab is going to be to measure the absorption spectrum of the chlorophyll that's in these green leaves up here, okay? Chlorophyll is a pigment that's essential for photosynthesis. And as we talked about earlier, light can either be absorbed or reflected. So I have a green leaf right here, and I know that plants must be doing what with green light? If it's green, this isn't emitting green light, it's actually reflecting it. And the lights in the classroom in here are emitting the full spectrum of light, right? All of Roy G. Biv is coming out of the ceiling here, but just the green is being reflected. So what does that mean? Does that mean chlorophyll is actually using green? No. If it's being reflected, it's not using it. What does that mean about the other colors? Well, it means that those must be absorbed by the leaf. But we want to find out how much. What do plants really need for photosynthesis? Do they prefer blue, violet, reds, oranges? Because we know that they don't really care about green. So how do we do this? Here is a beautiful California grape leaf. It's a native plant. It turns out though that all plants that have green leaves are loaded with chlorophyll. So what we need to do is we need to try to isolate the chlorophyll in this leaf. Because it's mixed in with other chemicals like beta carotene, which is an orange chemical that's in here too. So I can't really just, measure this directly. So here's what we're gonna do. We're gonna take a piece of chromatography paper, which is just a special porous paper that actually absorbs chemicals really easily, including petroleum ether, which is what we're going to put this paper into. And what that's gonna do is I'm gonna take this leaf, I'm gonna grind it into this paper along this line. And when I do this, all of the chemicals in the leaf are going to be pushed into the chromatography paper. You can see this green goopy line that I'm making here. I'm just gonna push it all in. Doesn't have to be pretty because it's... So I'm just gonna try to get all of this that I can in. I'm gonna use, the more I put in here, the better. So remember, what I'm grinding into the paper here is all of the chemicals that are in the plant leaf. Chlorophyll's going in here, but also other chemicals like beta carotene and other xanthanols and other cool chemicals that the plant needs. Again, all I care about here is the chlorophyll. So let's put it a little bit more over on this side. Yeah, that's amazing. Oh man, I got some up there, a little high, it's all right. All right, so I made this big line of plant juice, okay? It contains chlorophyll, but it also contains a bunch of other chemicals that I don't want, okay? So how do I separate the chlorophyll in here from the other chemicals, like the beta carotene? Well, what I'm gonna do is I'm going to put it in this jar. It's gonna have some petroleum ether in here. I'm wearing gloves because I actually don't wanna touch the chromatography paper too much with my hands. The oils in my hands could mess this up. And the line I drew on here was in pencil because graphite's kind of inert and it won't screw up my chromatography experiment. So what I'm gonna do is I'm gonna rotate this into a cylinder and staple it. So I wanna make sure that it's kind of even here. And kind of tricky. So staple it down there. I'm gonna staple it at the top. Okay, all done. So I've got this beautiful little cylinder here, okay? So now I'm gonna pour in some petroleum ether, and what's gonna happen is this petroleum ether is going to be wicked up by the paper, okay? Kind of like a sponge. If you put the end of a sponge into some water, the water is actually going to be pulled up into the sponge through capillary action, and that's what's happening with this chromatography paper. So what I'm gonna do is put on my safety goggles because I don't want to dump any petroleum ether into my eyes. And I am going to pour in a small amount of this petroleum ether into this jar. So I'm gonna put about 15 milliliters of this in the bottom, just enough to wick up into that paper. Okay, so I've got some in there. So what I'm gonna do now is I'm just gonna carefully set this down into the petroleum ether and let it wick up into the chromatography paper. I'm not gonna touch this. I'm just gonna put the lid on carefully. There we go. And now we're just gonna wait for about 15 minutes, and then I'll show you what this looks like. Yeah, you can actually see it wicking it up already. Look at how fast that's happening. Yeah, maybe you could. So you can see that there's a yellow band kind of forming on the top. That's actually the beta carotene. And then there are two different types of chlorophyll. We're not going to actually distinguish between the two, but you can see kind of a pale green band forming and a darker band forming on the top there. So it's already spreading out the different chemicals according to their densities and physical properties. So different chemicals will be wicked up at different speeds so that we can separate them.